Hi, welcome to the Metagenomics lab course. I'm Wenzi, a postdoctoral researcher from the MPAT research group. This is a tutorial video. Please feel free to contact me if you have any questions regarding the metagenomics section. You must have finished the preparation according to Wenjing Meng's introduction video. So now let's start the analysis directly. We will have 6 sections, plus a practical commands introduction in this tutorial. From the analysis part, I will guide you in importing sequence data, checking the raw data quality, quality control, building a phylogenetic tree, importing the database, and annotation. I will also attach the link from the QIIME official tutorial in the video. From this document, you can find more details about the analysis, and you may want to explore it yourself. Moreover, I summarized some practical commands for you in this plus section, with which you can improve your work efficiency, and you may also get an idea of how to troubleshoot errors during analysis independently. First, the QIIME platform is just like R. Any wrong typo fails the command. In this case, we recommend that you copy and paste the ready commands from the protocol we provided, but remember to adjust the file names and parameters if necessary. So here I have the first trick for you. For example, when you copy the code from your Word file, copy the content as you usually do. And just right-click Input, and you will get the content there. And if a command is running already, but you notice your code has some problems, and you don't want to wait until the error message, use Ctrl+C to terminate the command. If you want to enter any directory in PuTTY, use the command cd file name. And if you want to go back to the last level of the directory, use command cd space ... The command ll and ls will show you the content of the current directory and the access right of each file. Last but not least, if you don't understand a code, use command space -- help, for example. You don't understand the command for importing data, so write QIIME tools import help. This is the first command we will use for analysis. The command is for importing data. The basic frame of the code in the QIIME platform usually starts with a function. For example, the importing command has function QIIME tools import, then input and output. In this code, there is an extra parameter of type. Because there are both single-end and pair-end sequences. In our tutorial, we will use pair-end sequence data. The input in this command is a config file, where you describe the locations and names of your sequences, so QIIME can collect all your sequences for the following analysis. The output file, sample.qza, is a zipped file which includes all the sequences you guide QIIME to collect through config file. In this section, we use QIIME summarize function to check the summary of joined data with read quality. The code frame is the basic, with function, input data, and also output. By exporting the result of this step, you will get a directory named sample statistic. And check the 3 HTML file. And in the quality plot file, you can find two figures indicating the quality score of your samples from forward read and reverse read. This information is very important for the next step, quality control. Now, let's look at the figure together. First, we can see in the forward read for most sequences base, the quality score is over 25. But in around 190th sequence base, the quality score dropped lower than 20. This means the quality of this sequence base is quite poor. In principle, we keep the quality score of reads over 25 in the analysis. And if it's possible, we would like to cut all the bad quality sequences. And double-click the figure, you can see the quality score is only 11th at 194 sequence base. So let's plan to cut the forward sequence at 193rd sequence base for the quality control step. And now we look at the reverse read. We find the quality score drop lower 25 around 148 sequence base. However, we may be unable to cut the reverse sequence at 148 because we have two direction sequences and we need to merge both directions as one complete sequence. If we cut at 148, most of the sequences may not be merged and will be filtered out in the quality control step. In that case, we may need to cut at 211th sequence base. Here. And here is the evidence for our hypothesis. I tested cutting the reverse sequence at both 148th and 211th. In the first case, maximally 5% of sequences passed the quality control, but in the second case, over 90% of sequences passed the quality control. So it is okay to cut at 211th sequence base because only the tails of the plot dropped below 25 quality score, and the main body stayed over 25. And there is one condition we should be strict, is that the mean body of the plot dropped below 25. For example, for this project, we have to cut the sequence before 105th sequence base to keep the quality score over 25. As we decided the cutting point from the last step, let's just put the value in forward sequence cutting point at 193 and the reverse cutting point at 211th. In this code, you also say ptrim left F and ptrim left R. Those two are to trim the barcode or adapter from both direction sequence. But our raw sequences have gotten barcode and adapter cut, so we don't need to trim anything here. You will get three important files from this step. Feature abundance table, table. repsequence.qza, feature sequence file repsequence.qza, and also sequence after quality control stat data2.qza. So export the table QZA file to txt tab, and you will get the ASV table, which you will need for most of the following analysis. This one. A phylogenetic tree is a graphical representation that shows evolutionary history between a set of species or taxa during a specific time. We generate a tree in the analysis for phylogenetic diversity analysis. To generate a phylogenetic tree, we will use a lie-to-tree mapped fast tree workflow in the QIIME phylogeny plugin. Mapped is a multiple sequence alignment method. Multiple sequence alignment is the sequence alignment of more than 3 biological sequences, such as protein sequences, DNA sequences, or RNA sequences. In addition to the mapped method, there are also muscle, cluster W, and T-coffee method. You might notice in the output file there are one rooted tree and also one unrooted tree. The difference between a rooted tree and unrooted tree is whether a root node is specific. A rooted tree introduces an outgroup and has a root node, which can determine the direction of evolution, while an unrooted tree does not introduce an outgroup, has no root node, cannot determine the direction of evolution, and forms different classification units. Nothing needs to change in this command, so just run the command. To find out what species exist in our samples, we need to import a database as a reference. For the fungal project, download reference from Unite database and under QIIME release. And here are all the versions you can download. You need to import 2 files, a taxa file and also a FASTA file, which include taxonomy data and also sequence data, which you will use as reference to find out your taxonomies in your samples. These are the ready files we downloaded from Unite. And when you get both files ready, remember to upload it to the correct folder in your CSC project. For example, in our case, we run the QIIME analysis under Meta 2025, so we also need to upload the FASTA file and also taxonomy file under Meta 2025. And after you uploaded all the files, change the input file name with the file you downloaded. This one here, and also FASTA file here. And after you get everything is ready, you are ready to run the command. In the annotation step, we need a classifier to identify our samples. There are several classifiers, and you can even train your own classifier if needed. Today, we will use the classified consensus BLAST classifier. In this command, you also find a new parameter, p max accepts. This limits the maximum number of hits to keep for each query. BLAST will choose the first n hits in the reference database that exceeds per identity, similarity to query, and we only want to get the top one. The annotation may take some time. In my case, only 2 samples took about half an hour. After you get the output file, use the exporting command to get the taxa plot, bar plot folder. I have my folder ready here. And clicked the index.html file. And you can see an interactive interface where you can find a bar plot showing the frequency of taxonomy. This one is in domain level. And you can also find in other levels, for example, in the kingdom level, in the phylum level, and here is in the genus level, and also in the species level. And now you finish the basic microbiome marker gene analysis with QIIME tool.